Okay, as we get started on this lab, we are going to be finding the blood types for 4 different characters. So the first thing we're going to do is find the correct blood sample. So this is sample number 1. So sample number 1 goes with character number 1. Her name is Sarah Brown. And so what I'm going to do is I'm going to first put 1 drop of her blood in each circle. Now I'm going to put 1 drop of each antiserum. So I've got antiserum A, so that's antibody A, and I'm going to put antibody A with her blood there. Then I'm going to put antibody B in the next well that's labeled B, and I'm going to put antibody, they call it D, but it is the antibody against RH. Then I am going to stir these up and check for a reaction. And I'm going to zoom in a little bit more so you can see this a little bit more clearly. Now, if you look very closely, you can see that clumping there. So this means that it did react. So does that mean that there is A or is not A? That means that there is A. Okay. Let's do the same thing with B. I want you to notice I'm using a new toothpick now. I'm not going to cross-contaminate these. So for B, I'm going to stir this up. See if there's a reaction. And there is nothing. It looks exactly the same. I'm going to try the next one. RH. Going to stir this up. Ooh, and that is easy to see. It's been sitting for a while. That definitely reacted. Now, it doesn't have the same reaction as A, just because these are not actual blood samples. These are chemicals. And so, just the fact that it clumped, it's showing us that there is a reaction. The antibody had something to react with. And so, if it reacted with A, it reacted with RH, it did not react with B, I want you to think about what blood type that is. So, that is A positive. Go ahead and write that down for person number 1. Let's take a look at character number 2 now. We've got our blood samples and our antibodies already put into the wells. So if you notice, this one already has a little bit of clumps because the chemicals are a little bit old. So that's just dried chemicals in here. So other than those dried chemicals, there is no reaction here. So is A present? No. Take a look at B, and you notice there are some of those dried chemicals in this one too. But in addition to that, we do see that clumping there, and so there is B present. And let's take a look at RH. This one is definitely clumping. So if we saw a reaction with B and with RH, what blood type is it? Hopefully you came up with B positive. Excellent. Go ahead and write that down. Now let's look at our third character. So I already put the blood and the antibodies together. Let's stir this one up. And I definitely see that blood clumping there. You can see nice, neat clumping and agglutination. Let's try B. And I don't see a reaction with B. We might need to come back to that one. And RH, there's no reaction either. We'll come back to that one. There we go. With B, you are starting to see that reaction. You can start to see those clumping. So B is reacting. RH is not. So if we've got A, we've got B, we don't have RH. What blood type is that? Hopefully you came up with AB negative. Let's take a look at the last character. So I'll mix A. And again, I'm seeing just some of those dried chemicals in there. I'm not seeing a reaction here. Okay. move on to B. It's looking swirly, but I'm not seeing any clumping. So we don't have A, we don't have B. Let's try RH. You can already see RH is starting to clump. Beautiful. So RH is clumping, so we don't have A, we don't have B. What do we call it if there's no A and B? We call that O. And if there is RH, what do we call that? That is O positive. Go ahead and write that down, and now we'll take a look at how you'll complete the rest of the lab analysis. Okay, let's take a look at this. So our first question is, what blood types can be donated to each person? So what can they receive? So I want you to write this note here to yourself. You can receive the same proteins or fewer. You cannot receive proteins you don't have. So let's start with Sarah Brown. Sarah Brown has A positive. And so if we look at same proteins, well, obviously, she can receive A positive blood. Now let's look at this idea of fewer proteins. I want you to write on the side of your paper the 3 types of proteins, A, B, and RH. So she has A, she does not have B, I'm going to cover that up. She does have RH. So fewer proteins, let's take away one of those proteins. If I take away RH, she could take A with nothing. So what do we call that? A negative. Here's a shorthand how to write that, positive/negative. Okay, so let's go back. So we'll put RH back there, and let's take away A instead. So no A, no B. We're going to call that O. It has RH. It is O positive. If I take away everything, no proteins, that is O negative. Okay. So now the next question, Sarah Brown can receive from. So who of these characters can donate to her? So we see here A positive. Well, that's Sarah. That's herself. You cannot donate blood to yourself the majority of the time because if in an emergency you need blood, you don't have excess to give to yourself. If it was a pre-scheduled surgery in which the doctors know you will need a blood transfusion, you could plan ahead and donate blood to yourself. But generally, we can't plan ahead like that. So we're going to say no, Sarah cannot donate to herself. John cannot donate because he has B. Jim cannot donate because he also has B. Jennifer can donate because she does have O positive. She has the positive that Sarah has. She does not have anything extra. Go ahead and do the same for the rest of these people. Okay, now moving on, let's take a look at the back. So I want you to think about this remember up here. For the sake of Punnett squares, you do not have to consider positive and negative because that's a whole extra gene. That means we're going to do a dihybrid cross. That's a lot more complicated. We will get to those, but not yet. Otherwise, you must include those. So if we're talking about donations, use positive and negative. If we're talking about Punnett squares, ignore them. And so for these first 2 questions here, we are going to be looking at Punnett squares, so you may ignore positive and negative at first. So our scenario. These 4 orphans were raised separately and are trying to find their blood brothers or sisters from whom they were separated while very young. Jennifer Jones knows her father was type B. So notice they didn't include positive or negative because we're not using that information, so it doesn't matter. So we know that for certain. Now, if we looked back, we would see Jennifer is type O and John Jackson is type B. So let's take a look at this Punnett square in order to answer the question. So answer the question in a complete sentence. Explain your answer in a complete sentence by telling what alleles the dad and mom could have to have produced children with these blood types. Support your answer with a Punnett square. So if the dad is type B, I know that he has IB. I don't know his other allele. So we're just going with what we know for certain. So I'm going to write the IB, leave that blank. Now, Jennifer has type O blood. I know she has to be lowercase I to have that type of blood. John Jackson is B, and again, all I know is IB. I don't know his second letter, so I'm going to leave that blank. And so if I look here on this Punnett square, can I fit any people in? Well, I'll fill in what I know, which isn't a lot. I know this much. And this right here could be John. So that's possible. Now, I don't know this letter, but I do know that Jennifer is his daughter. So if he has a lowercase I, then it's possible that these 2 children have the lowercase I. And if this mom has a lowercase I, then we could have Jennifer right there. So yes, it is possible for both of these to be siblings, having this same father if the dad has these two alleles and the mom has this allele. So, answer your question. Could they have the same father? Tell what alleles we know the mom and dad have. Then when you get to question 2, it's now asking, could all 4 orphans be siblings? So we already know that it's possible for John and Jen to be siblings. Start from here, copy that here, and then try to add in Sarah and Jim and see, is there any possibility for all 4 to be siblings, and what would the 2 parents' genotypes have to be for that to be the case? Question 3 is a lot like what you answered on the front, but needs an explanation. Questions 4 and 5 are a lot like what you did on Punnett Squares number 3, so hopefully everything on the rest of this is easy. Of course, ask if you need any support.